Right, this is going to be a mini lesson on enzymes and enzyme regulation. So this will be the second part of chapter 6. So what are enzymes? Well, enzymes speed up reactions. They are known as protein catalysts. So enzymes, they're going to increase the speed of a reaction. Now, something that's very interesting about enzymes is that they are not altered themselves. So when they're involved in a chemical reaction, they catalyze a chemical reaction, they do not get used up in that chemical reaction. So they can be recycled over and over again. And how they speed up a reaction is that they're going to decrease what's called the activation energy for a reaction to take place. So let's take a look at an example. So if you take a look at our figure down below, we have a reaction without an enzyme and a reaction with an enzyme. Now, in the previous lesson, I talked to you about exergonic reactions that are spontaneous and they don't require any free energy input. Okay, well, they're not exactly spontaneous in the sense that it's going to happen quickly on its own. It will eventually happen, but we got to speed it up. Okay. So, for example, if you have a piece of wood and you are going to, have it a log in a fireplace or whatever, it's going to take a long time for that log to convert to something else. Okay. It'll happen. It'll just take a long, 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 long time. So there's this activation energy barrier that's in place. Okay. If we lower that activation energy, such as by adding a spark, like from a match, or maybe an enzyme, then the energy barrier is lowered. So that activation energy will get the reaction going and it speeds it up. Okay. So exergonic reactions, they do occur, but sometimes they need a little push to get that activation energy barrier lowered so that they can be sped up. Okay. So enzymes are the ones that lower that energy activation. Okay, so they're going to speed up the reaction by lowering that energy activation. Okay, so we can see here that you take a look over with the yellow arrow over to the right, the change in G, the change in free energy is unaffected by the enzyme. Okay, so this has nothing to do with the free energy. So we're not adding any energy into this this reaction. We're not taking away any energy in this reaction once it's started. So the only thing that the enzyme is doing is just lowering the amount of energy needed, the activation to get it activated. Okay, so it actually doesn't add or decrease any energy. So how does it do that? Well, it has to do with the structure of the enzyme. So enzymes are basically, they have a structure to them that is very specific to receive a certain substrate. So if we take a look at our diagram of our enzyme, the enzyme structure has what is called an active site. And that active site is where the substrate or the reactant in the chemical equation is going to bind to that protein. And it's a very specific active site that can only bind with a specific substrate. So that enzyme can only have one type of substrate bind to it. It cannot have a different substrate bound to it. So for example, the enzyme lactase can have the disaccharide lactose bind to its active site. It cannot have any other type of disaccharide attached to it. Okay, sucrose will not attach to the active site of lactase. Okay, so the active site will only bind to a specific substrate. Okay. And once we have the substrate attached to the active site of the enzyme, we form an enzyme substrate complex. So here is an example. We have our enzyme. That enzyme has a specific active site where the substrates will bind to that active site. And during that process, those substrates are so close together that they can have that reaction take place. Okay, so the substrates then can be converted to whatever product, and then they get released. Now, the shape of the enzyme, or the enzyme itself, is not dissolved or reduced in any form. So once the products are released, the active site becomes available for new substrates to bind to it. Now, with enzymes, certain environmental factors can affect the enzymes. So we talked a little bit about proteins becoming denatured in our macro molecule talk in chapter 3. So you should already be aware that proteins, they can change shape when the environment changes surrounding them. And that's going to cause them to become denatured. And so they can be no longer functionable. Since enzymes are proteins, a protein or an enzyme can be denatured if there's a change in the environmental conditions. So certain enzymes have certain optimal temperature ranges in which they can optimally catalyze reactions. So if the temperature gets too low or too high, that could change the shape of the enzyme, making it no longer be able to accept a substrate on its active site. Okay. This can also happen if there's a change in the salt concentration or the pH. There's enzymes typically, they have a certain pH range that is optimal for them. And if the pH is too high or too low outside of that optimal range, then they become denatured and they no longer work. Certain chemicals can also cause that to occur. So here's an example of two different enzymes with two different optimal temperature ranges. Okay, so you can see that there is a range to it, but if the temperature is above 60 degrees in the one on the left, then that enzyme's not going to be able to function anymore. Same thing with the graph below, we have pH, optimal pH for certain enzymes. In our body, we have a lot of different enzymes, but in our digestive tract, we have a stomach enzyme that its optimal pH is 2. Okay, so once that stomach enzyme gets moved into the intestines, the small intestine, the small intestine's pH is a lot higher. Okay, it's closer to 7 to 9, pH of 7 to 9. And so it's going to denature that stomach enzyme once it gets into the intestines. Okay, whereas the opposite, okay, trypsin is an intestinal enzyme. Its optimal pH is around 8, okay, so weak base. And if, for example, too much acid spills into the intestines without any buffers, then it can cause the denaturization of trypsin. So that we have an optimal pH range for our different digestive enzymes. Okay, so as far as enzymes go, sometimes we have, in order for that, what's called induced fit, where the enzymes kind of hug the substrates and have the reactions take place, we might have some helpers that help the whole process. So a cofactor is a non-protein enzyme helper. So it helps the protein enzyme. So our cofactors may be inorganic or organic. If it's organic, we call it a coenzyme. So if we think about our diet, it's mainly just a bunch of macromolecules, right? We have carbohydrates, we have proteins, and then we have lipids. And then, anytime we eat any organic, food item, it's going to have nucleic acids in there because it's made of cells. But what about the vitamins and the minerals? We always say, oh, we got to get enough vitamins and minerals. Well, the vitamins and minerals, they're involved with, they're the cofactors of our enzymes. They're the helpers of our enzymes. So most vitamins that we consume, they act as coenzymes. And the minerals can act as a cofactor, an organic cofactor. So, think of like vitamin K or vitamin B6. Okay, so those we get from our diet, and they're important coenzymes. Um, some of the minerals, like we have to have a little bit of zinc and so forth. So that would be an inorganic cofactor. So those make up some of the vitamins and minerals. And we just need minute quantities of them, right? Because they can get recycled. So checkpoint, explain how an enzyme speeds up a reaction. Okay, so what does the enzyme do? It provides an active site in order for the substrate to bind to it. So that allows the substrate to have basically a surface area in order for a chemical reaction to take place. So that will, as that enzyme undergoes that induced fit, it kind of hugs that substrate at the active site. It's going to push the molecules together or pull them apart so that the reaction can occur. So that's lowering the activation energy in order for the reaction to occur. So sometimes enzymes, they don't work like they're supposed to. And that could be due to some sort of chemical or some sort of toxin or a poison or something like a pesticide or an antibiotic. And so we call those examples inhibitors. So they will inhibit the function of that enzyme. So we're going to talk about a couple different types of inhibitors. The first one is called a competitive inhibitor. So what a competitive inhibitor does is it binds to the active site of the enzyme. And so the substrate can no longer bind to it. Okay, so you can see in our picture below, at the active site of the enzyme, this blue competitor somehow managed to bind to it instead of the substrate, the green guy. Okay, and so now the substrate cannot bind to it. So the reaction cannot occur for the substrate. In a non-competitive inhibitor, the inhibitor does not bind to the active site, but it will bind to another part of the enzyme, causing the enzyme to change shape. And as it's changing its shape, the active site changes too along with it. And so the substrate can no longer bind to it because the active site is now different. So non-competitive, it's not actually competing with the substrate, but it's changing the enzyme to prevent the substrate to bind to it. So here's a question. What determines whether enzyme inhibition is reversible or irreversible? Actually, we didn't really talk about that. So let's talk about this first. Okay, so cells switch genes on and off that encode for specific enzymes or regulate the activity of enzymes. So in our body, we might not have competitive or non-competitive inhibitors inhibit the function of our enzymes. Sometimes it's just due to the fact that we don't need any more of a certain product being produced, so we need to shut that enzyme's activity down. So allosteric regulation can either inhibit or stimulate an enzyme's activity. Okay, so it kind of acts like a competitive inhibitor or non-competitive inhibitor, but it's regulating the ability for that enzyme to function. Okay, so we have our genes, they switch off and on to encode for specific enzymes or regulate the activities of those enzymes, right? So in that case, the regulatory molecule will bind to a protein at one site, affecting the protein's function at another. So remember, this would be like an enzyme. So we can have a regulatory molecule that's produced by a gene, for example. It's going to bind to the enzyme at one site, and that makes it so that the proteins or the enzymes that doesn't function. So it could either cause it to function or the enzyme to not function at all. So here's an example. We have our alloteric enzyme. We have the enzyme, we have the activator, and then if the activator is present, it's going to create a stabilized form for that enzyme to function. So the active sites are ready to go. If the inhibitor is in place, then the enzyme's active sites are closed up in that case, so it's not going to allow the enzyme to work. Yeah. So cooperative, I can't even pronounce the word. It's a form of alloteric regulation that can amplify an enzyme's activity. So you can see how the substrate, it's basically going to cause, it's going to bind to one site, and that's going to affect the other active sites to change as well. Okay, so we have the alloteric regulation that can amplify an enzyme activity. So the substrate is binding to one of the active sites, and that affects the reaction in different active sites. And then we can have some sort of metabolic pathway that will shut down due to feedback inhibition. So, for example, at the beginning of chapter 6, we talked about metabolic pathways where we might have one enzyme causing a reaction to occur to form a product, and then we have a different enzyme come along and take that product and react and cause a new product to form, and then another enzyme to come along to take that new product to cause it to react to form a final end product. So we ended up with like 3 enzymes undergoing back-to-back reactions. But eventually we have enough of the end product, so we got to shut it down. So in feedback inhibition, the end product shuts down the metabolic pathway, okay, through feedback inhibition. So we can see here that at the beginning, we have our first enzyme involved, and that first enzyme is going to have an open active site for a particular molecule to bind to it, a substrate. And then a reaction will form to form the product. In that case, it's intermediate A. And then another reaction will occur with a different enzyme to form the product intermediate B. And then another enzyme to form another product called intermediate C. And then another enzyme forming another product, intermediate D. And then finally, another enzyme producing the end product, isoleucine. Now, let's say we have this reaction going on and on and on, and we end up with a buildup of isoleucine. Well, eventually we're going to have so much isoleucine that we don't need any more. So when that occurs, the isoleucine binds to the allosteric site of the original first enzyme, enzyme number 1, and changes its shape so that the active site is not available for the reactant to bind to it. So it's going to shut the whole metabolic pathway down. So that's called feedback inhibition. So that's it for our mini lesson.